Hi guys, welcome to my YouTube channel. Don't forget to subscribe and share.
Hai, Assalamualaikum warahmatullahi wabarakatuh. In this video, I would like to explain about chapter 1.4 protein. Okay. So now we have done discuss water, carbohydrate, lipid, so that we proceed to protein. Okay, this is the learning outcome. Okay, explain how amino acid are grouped, describe formation and breakdown of dipeptide, explain primary, structure, secondary, tertiary, and quaternary level of protein and types of one block, explain the effect of application temperature on the structure of protein, and also classify protein according to their structure. Okay, so what is protein? Protein is a large complex molecule composed of amino acid which link together by peptide 1. So mean here, protein adalah satu molekul yang besar dan it is made up of the monomer called amino acid. So amino acid and amino acid is linked together by bond called peptide 1. So this is an example of the protein, or we call polypeptide. Why polypeptide? So it means that it is composed more than 2 of amino acid. So this is amino acid 1, 2, 3, 4. So in the diagram, you have 4 amino acid that link together by the peptide bond. So we call this structure as polypeptide. So now let's take a look at the basic structure of amino acid. So here is the basic formula. Okay. So from the basic formula, the amino acid can be we can class from the basic formula as 3 functional group and double NH2. This is represent amino group. Okay, and then these are represent residual chain, or we call it side group. Okay, so these residual chain or side group, these will determine the class of amino acid. Okay. And then this is C double bond OH. So this is represent carboxyl group. Take a look and please remember this structure. Okay, the basic structure of amino acid: NH2CH, C double bond OH. The functional group is amino group, R group or side chain, and this is carboxyl group. So, as I mentioned, this residual chain is the important to determine the group of amino acid, because amino acid can be grouped into 4 groups. Okay, non-polar, polar, acidic, or basic. So all these is referred to the R group. For non-polar amino acid, okay, let's take a look for polar amino acid. Polar amino acid, it will has a polar side group, means it will have slightly charged or partially charged at the R group. So example here, okay, look at this, they have the hydroxyl. So the hydroxyl will have a slightly negative charge. So here also, because due to the hydroxyl, then okay, so means that if the R group has slightly or partial charge, so it will cause the amino acid is grouped as polar amino acid. Okay, next is non-polar amino acid, in that the amino acid at the R group has no charge, no partial charge. So the amino acid with the R group with no charge, for example here only consists of carbon and hydrogen. This is carbon ring, so no charge. So the amino acid is grouped in non-polar amino acid. Next, the third one is acidic amino acid. So here is the R group, and then if the R group has a negative charge, so the amino acid is called acidic amino acid. Is grouped into acidic amino acid. Okay, and number 4, basic amino acid. Look at the R group, this residual chain, and then yes, you can see this is a positive charge. Okay, the positive charge at the R group. So this amino acid will be grouped under basic amino acid. So I repeat, we have 4 amino acid, which is non-polar amino acid with no partial charge. And then we have polar amino acid, mean that at the R group has slightly charge or partial charge. And then the acidic amino acid at the R group has negative charge. And basic amino acid at the R group has a positive charge. So here, the example for non-polar, polar, and then for the acidic and basic amino acid. No need to memorize all the name of amino acid. Just know how to identify the group of amino acid. Okay, now let's take a look at the formation of peptide bond. So this is represent one molecule of amino acid, and this is the second molecule of amino acid. The formation of peptide bond involves condensation process. So condensation process same goes to formation of maltose, formation of triglyceride. The condensation means involve removing of water molecule. So removing of water molecule, for the first amino acid, the hydroxyl group is lost from the carboxyl group. Okay, so the hydroxyl is removed from the carboxyl group, but for the hydrogen, it is removed from amino group. Okay, so as we know, amino acid has three functional group. This is called amino group, this is called R group, and this is carboxyl group. So the removing of water molecule involves carboxyl group, which lost the hydroxyl, and then the amino group, which lost the hydrogen. So the process is condensation, and then the product is called dipeptide. Okay, the product is dipeptide. So here we can see this is the first amino acid, and this is the second one. So the carbon and nitrogen is linked together now. So this is called peptide bond. The bond between carbon and nitrogen is peptide bond. Okay, now let's take a look. Okay, in this structure, okay, as 1, 2. Okay, so means the structure given is dipeptide. So here is peptide bond, and then this peptide bond is react with another one molecule of amino acid. If you look out at this equation, don't worry, because all the R group is not involved in the reaction. The reaction only involved the carboxyl group for the first one, and then the amino group for the second amino acid for second structure. Then show removing of water molecule. The arrow write down condensation. So now the bond is formed between this carbon and this nitrogen. So now they are linked by peptide bond. Remember, the side chain or the R group is not involved in the reaction. Okay, so here similar, same thing. Okay, this is methionine that react with aspartate. No worries, you can do it. Okay, and then in this diagram it show that 4 amino acid actually react to form polypeptide. Don't worry, because as I have mentioned, the side chain is not involved. So for this one, okay, we lost the hydroxyl group. This lost the hydrogen atom. This one lost the hydroxyl group. This one lost the hydrogen. This one lost the hydroxyl group, and this one lost the hydrogen. So how many peptide bonds form? 1, 2, 3. Remember, peptide bond is the bond between carbon and nitrogen. So the carbon and nitrogen, the carbon with double bond. Oh, so here 1, 2, 3 peptide bond. Then breakdown of peptide involve hydrolysis reaction, means the process involve addition of water molecule. So this is structure of dipeptide. And when one molecule of water is added, the carbon here will get back the hydroxyl group. So carboxyl group is complete now. And then for the second amino acid, the amino group will get back the hydrogen atom. So that now we have 2 molecules of amino acid. So this is the basic formula to show the process formation of dipeptide and breakdown of dipeptide. Yep, I repeat, for the formation of dipeptide, it involves 2 molecules of amino acid, and the process is condensation, removing water molecule. The water molecule is removed, involve carboxyl group which lost the hydroxyl, and then the amino group which lost the hydrogen atom. Okay, and then the product is called dipeptide. The bond that link 2 amino acids is called peptide bond, and the side product is also one molecule of water, not shown in this diagram. Okay, then process of breakdown of breakdown of dipeptide, okay, involve process hydrolysis, which one water molecule is added to break the peptide bond. So the product is two molecule of amino acid. Now is level of protein structure. So we have the primary structure. Look at the structure, and then the secondary structure, tertiary structure, and quaternary structure. So the structure is depend on structure of polypeptide itself. So for primary structure, it only consists of peptide bond. Why? Because primary structure, the polypeptide arranged in linear sequence. So this is represent one amino acid. So the amino acid form the polypeptide, and the polypeptide is present in linear sequence. So on the peptide bond present in primary structure. Example is in insulin. Okay, for secondary structure, okay, we have two common structure, which is alpha helix and beta sheet. Now, the polypeptide is not linear anymore. In alpha helix, the polypeptide is coiled. So, it coiled and form helix structure. So, to maintain the helix structure, another one accessory bond here, this is hydrogen bond. Beta sheet, the polypeptide sheet is folded. Okay, for alpha helix, the polypeptide is coiled, but for beta sheet, the polypeptide is fold, berlipat. Ini dia okay, macam wayar telefon tu. So to maintain the structure of alpha helix and beta sheet, okay, other than peptide bond, the structure is maintained by hydrogen bond. Okay, next, tertiary structure. So now look at this diagram. The polypeptide is coiled and folded at the same time. So the structure of polypeptide that coiled and folded, so this is represent tertiary structure. So the shape is held firmly in position by the interaction between side chain of the various amino acid. So we know polypeptide consists of many amino acid, and the structure of each amino acid must have their side chain. So the interaction between the side chain will interact each other and will help firmly the position of tertiary structure. So to maintain this shape, this is the bond, okay, and the bond is formed, okay, between the side chain of the amino acid. We have hydrogen bond, ionic bond, disulfide bridge, van der Waals forces, and hydrophobic interaction. You need to remember all this. Okay, tertiary structure is maintained by this bond: hydrogen bond, ionic bond, disulfide bridge, van der Waals forces, and hydrophobic interaction. Example is myoglobin. Last, okay, is the quaternary structure. So quaternary structure, if you can see here, okay, different color, yellow, orange, green, blue. So these represent different polypeptide. Look at the structure of each polypeptide. They are coiled and folded. So means they are in tertiary structure. But the polypeptide, the different polypeptide, they are pulled together. So this is quaternary structure. Means the protein consists more than one polypeptide chain. So example is collagen. Okay, collagen has a fibrous protein consists of 3 helical polypeptide that is supercoiled. Okay, and then hemoglobin, which are globular protein, which 4 polypeptide subunit. So again, okay, this is collagen. We can see 1, 2, 3. So 3 polypeptides are together. Okay. Then for hemoglobin, it's 1, 2, 3, 4. So 4 polypeptides are together to form hemoglobin. Okay, so here primary structure, if you can remember, only peptide bond present. But for secondary structure, peptide bond, hydrogen bond. And for tertiary structure and quaternary structure, the structure is maintained by another 5 excessive bond, which is hydrogen bond, ionic bond, disulfide bridge, van der Waals forces, and hydrophobic interaction. And now it's about classification of protein. Protein can be classified into 3 groups according to their structure: conjugated protein, fibrous protein, and globular protein. Conjugated protein is referred to protein which associate with non-protein. Remember, conjugated protein is referred to protein that associate with non-protein. So that is conjugated protein. Example is glycoprotein. So glycoprotein is a protein that mixed with carbohydrate. Okay, lipoprotein, protein mixed with lipid. Hemoglobin, protein with the iron phosphate group. And then globular protein. Globular protein is referred to the polypeptide chain folded into a compact spherical shape. So if you can see, the structure of globular protein is like a ball. Dia lebih kepada macam benang yang bergumpat. So this globular protein soluble in water. Why soluble in water? Remember, it consists of hundred of amino acids. So at the side chain, maybe the amino acid is polar amino acid, acidic or basic. So they may have charge. So when it reacts with water, so water can dissolve the polar compound or the ionic compound. So this globular protein important in metabolic reaction. Example is enzyme. And then the fibrous protein. So you can see here for the fibrous protein, look at the structure of polypeptide. The structure is long chain. Okay, it's not compact. So this fibrous protein is insoluble protein. Okay, globular protein is soluble, but for fibrous protein is insoluble. Then the importance of fibrous protein, it will act as structural molecule. Example is keratin, collagen. So that's why for those yang memang nak kulit tegang, dia akan ambil collagen. Sebab apa? Strukturnya adalah untuk bagi additional strength, lurus. Okay, so this is the difference between fibrous protein and globular protein. Right now, let's proceed to the factor affecting structure of protein. So, have 2 major factors here, which is pH and temperature. For pH, extreme pH can cause protein loss its conformation. Okay, why? Because it will break the bond that cause the 3D shape to be changed. Okay, remember, okay, for the amino acid, okay, it can be acidic or basic amino acid. So, for example, the acidic amino acid replaced in solution yang condition yang berakali. So, mesti ada berlaku interaction antara acidic dengan basic. So, reaction itu will cause the bond in the polypeptide is being break. So, bila bond pada molekul itu terputus, apa yang akan berlaku? Terputuslah bond-bond excessive tadi. For example, hydrogen bond, then van der Waals forces, hydrophobic interaction, disulfide bridge, ionic bond will be break. So the structure, the tertiary structure or the globular structure will be lost. Same goes to the temperature. Okay, when the high temperature, when the high temperature, protein will lose its conformation. Why? When high temperature will cause the non-covalent bond will be break, especially hydrogen bond, because hydrogen bond is weak bond, easy to be break and reform. So when high temperature, when the hydrogen bond is break, so it cannot maintain the structure of protein. So when protein lost its conformation, we call the protein is denatured. Okay, denatured is referred to protein lost its conformation and cannot function. So that is denatured. Okay, and then after the protein denatured under physiological condition, it can be reversed. So this is called renaturation. For example, kalau kita dah tahu dekat rambut kita tadi kita ada fibrous protein, rambut katakan rambut kita lurus je, and then kamu pergi buat perm. Okay, lepas tu kamu buat perm, and then dalam beberapa lama dia akan balik kepada lurus. Ataupun orang yang berambut keriting, and then dia pergi buat rebonding. So dia memaksa untuk the polypeptide chain to be strict. Tapi at a long period of time, apa akan terjadi pada rambut tu balik? It return back to the original. Okay, kalau dia keriting, dia akan kembali kepada keriting. So itu adalah denaturation. So that's all for protein. Bye.